and welcome to this next video in OCR A Level Biology. We're still on Module 2.2, which is Biomolecules, and this is the last video in this module. So, this last video is going to be about just finishing off proteins and then talking about some inorganic ions as well. So, let's start. So, let's pick up where we left off. So last time we talked about, in the last video, we talked about how different amino acids can be bonded together, can be joined together to form a dipeptide. So here we have two different amino acids being joined together. They all come together. In the process, they release water. This is known as a condensation reaction. And then a peptide bond is formed between the two amino acids. So this is the peptide bond here. It's between the O and the N. That's what we term the peptide bond. And here you can see water is being released in the process. But also, so that's making a dipeptide. We also spoke about in the last episode, in the last video, that you can hydrolyze this peptide bond. So you can break this peptide bond and get your two amino acids back by a reaction known as hydrolysis. This requires water. So here, hydrolysis requires water be put into to be split. So it requires water to be split, and an OH will come here and the H will come here. So that's hydrolysis, going from a dipeptide to two different amino acids. But now we're going to talk about levels of protein structure. So let's get started. So protein structure. So protein structure. So we have four levels of protein structure, one of which, the first one being the primary structure. And the primary structure is simply the sequence of amino acids. So the primary structure is simply the sequence of amino acids. So the sequence of amino acids in a protein chain is called the primary structure. And so it's dependent upon the number of amino acids, but also what order the amino acids are in. As this primary structure then determines all the other levels of protein structure. And hence it can determine a protein's function. So here we have different proteins. So I'm going to use red. So here we have the different amino acids being bonded together. So here if we say that the primary structure is just, so this is what we term the primary structure. So these circles here, these are different amino acids being held together by peptide bonds, and this is what we term the primary structure. Now we're going to move on to secondary structure. So secondary structure is where we're going to have the protein chain, the amino acid chain, is going to start to fold in on itself. And this will happen via hydrogen bonds. So we have the initial folding of the protein, and this can be done using, and this is done using hydrogen bonds. So hydrogen bonds are used to create the secondary structure or the initial folding of the protein. Let me just scroll down. And then those hydrogen bonds can form in one or two different structures within to form the secondary structure. We either have an alpha helix, and an alpha helix will look like something like this, basically like a spiral, so that's an alpha helix, or a beta pleated sheet. And a beta pleated sheet will look something like this. So this is the alpha helix, and this is the beta pleated sheet. Now in the alpha helix, you can see here how there are hydrogen bonds being formed here, hydrogen bond being formed there, hydrogen bond there, and hydrogen, and so on. In the beta pleated sheet, hydrogen bonds are being formed like this. So these blue dashed lines that I'm drawing in right now, those are the hydrogen bonds that hold the amino acids together in one of these two structures. So that's secondary structure. Tertiary structure is something a bit more complicated. Now remember, these hydrogen bonds are forming within like the main backbone of the amino acid chain. What do I mean by backbone? Well, if we take a look at a typical amino acid, which is this one, we can see that the backbone, the backbone here is this section here. So this section is the amino acid backbone. And it's the hydrogen bonds that occur within the backbone only. So we don't have any mention of the R groups yet. The R groups come into play in the tertiary structure, which is what we see here. So this is the tertiary structure. Tertiary structure. So this is involvement of R groups, and it's the further folding of the protein. Of of the protein. So the tertiary structure when these alpha helices and beta beta sheets start to again fold. So this is causing more further folding of the protein to form the tertiary structure. So here a number of different bonds are used here. So a number of different bonds are used here. We have first of all ionic bonds. Second, we have something called disulfide bridges, which can only occur in amino acids which have sulfur. So cysteine, for example, which has sulfur in, disulfide bridges or disulfide bonds can occur between them. We also have hydrogen bonds, and which have also has something called hydrophobic and slash hydrophilic interactions. So we have hydrophobic and hydrophilic interactions, and that we can be and this can all of these four bonds can be seen in this diagram here. So let's zoom in on this diagram here. So here we have a diagram of a protein that's folding into its tertiary structure. So let's go through the different bonds that we mentioned. So these ones, so we have an R minus and an R plus. This one, which is here, this one is called the ionic. These are the ionic bonds. Here we have two sulfurs being joined to each other. This is the disulfide bridge or disulfide bond. But disulfide bridge is a bit more technical for me. So disulfide bridge. Here we have hydrogen bonds. So when a hydrogen bond, a slightly positive hydrogen binds to a slightly negative oxygen, right there. So that's a hydrogen bond. And also here we have hydrophilic and hydrophobic interactions. So the red bits are amino acids with hydrophobic R groups. So let me zoom in on here so I can write it neatly. So the red bits that we can see here are the amino acid with hydrophobic R groups, and the dark blue ones that you can see. So those hydrophobic R groups, those amino acids with hydrophobic R groups are going to be folding on and remain on the inside, whereas the blue ones, which are the amino acids with hydrophilic R groups, they are the ones which are going to be remaining on the outside because they are within the water soluble environment. Because the hydrophilic amino acids love water, so they can be attracted to water. But the hydrophobic amino acids, because they are water hating, they are going to remain away from water, so they're going to be on the inside of the protein. So that's tertiary structure. We also have one, so most proteins stop at tertiary structure. Well, most proteins. Some proteins stop at most proteins stop at tertiary structure. But there are a few proteins that go beyond tertiary structure. One of them being hemoglobin, and hemoglobin can actually go into something called quaternary structure. So quaternary structure. So the hemoglobin is one of those examples of proteins that have a quaternary structure. So quaternary structure involves one or more than one polypeptide chain. So these are more than one polypeptide chains that are joined together by various interactions, and they often have extra bits added on. Yeah, so quaternary proteins, such as hemoglobin, which we will talk about here, have more than one polypeptide chain. An example of this being hemoglobin, when I think was collagen, and hemoglobin is an example of a globular protein. Globular proteins. And it's also known as a conjugated protein. Conjugated. And what this means is that a conjugated protein is a protein that contains some sort of heme group or an phosphatic group. So a conjugated protein has a phosphatic group attached to it. In hemoglobin, it's the heme group. So let's talk about hemoglobin first. So this is hemoglobin. So we're going to zoom in to the hemoglobin. So we're going to zoom into hemoglobin here and have a look at the structures associated with it. So the first one, so hemoglobin is a protein that carries oxygen, that transports oxygen in your red blood cells around the circulatory system, around the cardiovascular system. So hemoglobin is made up of 4 polypeptide chains. So here you can see here are 2 polypeptide chains and here are another 2 polypeptide chains. These 2 polypeptide chains are called alpha units, alpha globins, alpha globin. So here's one alpha globin, here's the other alpha globin, and then the bottom two are called beta globins. So here's a beta globin, and this is a beta globin. So but two alpha, so hemoglobin is what we call a tetramer. So it's a protein made up of 4 polypeptide chains, 4 polypeptide chains made to the tetramer. In the case of histones, for example, histones, which are proteins that surround DNA, they are called optimers because they are made up of of 4 dimers, so 4 sets of 2 proteins to form an optimer, 8 proteins in total. But we don't need to go into that much detail about histones, we're just focusing on hemoglobin. You can also see that hemoglobin contains a heme group. So this is the heme group here. So here we have the heme group, and that heme group contains iron. And it's the oxygen that binds to the iron within the heme group. So one oxygen molecule can bind to one phosphatic group or one heme group within one hemoglobin molecule. So that means in total, one hemoglobin molecule can carry up to 4 oxygen, 4 O2 molecules. So in theory, so in fact, that will carry up to, because O2, oxygen, it will actually carry up to 8 oxygen atoms or 4 oxygen O2 molecules in one hemoglobin molecule. And you have thousands of these, even millions of these, so you can carry up to like, what, 4 million O2 molecules? Something like that. But anyway, I digress. So that's hemoglobin, an example of a quaternary structure, globular conjugated protein. But we also have other proteins as well, which I would like to mention here. So we're going to draw up a nice table. So conjugated, no, not conjugated, fibrous and globular proteins. So here is that table. And we're going to have here fibrous on the left hand side, fibrous proteins. And here we're going to have globular proteins. So we're going to look at some differences between fibrous and globular proteins. So fibrous proteins, they are regular repetitive sequences. I'm going to just steal from the textbook. Regular, repetitive, not repetitive, repetitive sequences. They are regular repetitive sequences, that is very hard to say, amino acids, and they are usually insoluble in water. So they're insoluble in water. And because of these features and the name, they help to form fibers, so they can form fibers, hence the name fibrous proteins. And they tend to have a structural function. One example of this is collagen. So there are multiple examples for this, so I'm going to do purple examples. So collagen is it's the most abundant protein in your body. Collagen, keratin, which helps to form keratinocytes, and which is found in the skin and in hair. And keratin is actually rich in cysteine, the amino acid cysteine. So there's lots of glutathione disulfide bridges forming between the polypeptide chains. And this makes keratin very strong and it's very suitable for animal horns. So goat horns are mainly made out of keratin. Hair as well. Hair is mostly made out of keratin as well. Skin has the top keratin in it as well. Nails, fingernails, toenails, they all contain keratin. And lastly, we have something called elastin. And elastin is a very extensible, very flexible protein. So because of this cross-linking and coiling within the structure, within its structure. So it's strong and extensible. So it's also found in our skin. And in the lungs, elastin is found in the lungs to help them expand and contract. And also, elastin is found in blood vessels such as arteries and veins to allow them to be able to stretch and recoil to help maintain the pressure of the blood that is pumped through them and to be able to withstand the pressure as well. So globular proteins, so that's fibrous proteins. So globular proteins are a bit different. So they tend to roll up into a spherical shape. So they have a spherical, I'm just checking the spelling, is spherical shape. So they do have hydrophilic groups, hydrophobic inside and hydrophilic are out. So they can be soluble in water. So the protein is water soluble. So they often have very specific shape and they can take up various amounts of roles. So they have various roles as enzymes, so such as pepsin, so enzymes, hormones, so insulin, I think glucagon as well, hormones, adrenaline also, and also transport transport as well, such as hemoglobin. So the examples we talked about are hemoglobin, insulin, which is a protein hormone used for regulating blood glucose levels, and pepsin. Pepsin is an enzyme that digests proteins in the stomach. Okay, so that was everything about proteins. So now, quickly, I'm just going to go into before we finish up, I'm going to talk about the inorganic ions that are necessary for life. So we're going to talk about inorganic ions very briefly to finish off. So here we're going to have inorganic ions. So I'm going to mention a couple of these inorganic ions. But if you want more information about the inorganic ions, go to pages 72 and 73. On my textbook, it's missing the stuff on page 73, so I've just written the stuff in. But your textbook might have. Actually, I'm going to go into the cations first. So cations, these are positively charged ions. So cations, so just to name a few, Ca2 plus, and that is used in rigidity of bone, maintaining rigidity of bone, teeth and cartilage, important in clotting blood clotting, and also in muscle contraction, stimulating in muscle contraction as well. I'm just going to mention two cations. Contraction as well. And then we have another cation, let's say hydrogen, H plus ions. And this is involved in respiration, photosynthesis, but it's also in transport of O2 transport, and it's also in regulation of pH, so pH regulation of pH in the blood. And very briefly, so those are just two cations that you need to know. I'm not going to do all the cations. So anions, I'll do the ones that are listed here that are not listed in my textbook because they might be the same for your textbook. Some of the textbooks apparently don't have them. So let's do no, not chloride, because chloride's massive. Phosphate, so PO43 minus. So phosphate, this is used to increase bone rigidity, and supports teeth. It helps in root growth in plants, regulates, it's a component of phospholipids, is used in ATP production, and phospholipids. So that's PO43 minus, and hydroxide OH minus is involved in blood pH regulation. And that is it. So that is everything for module 2.2, which is biomolecules. So thank you very much for watching. Hope you found this useful. In the next video, we're going to start module 3, which is exchange and transport. In the next video, we're going to start with module 2.3, which is all about, okay, let me get this right. In the next video, we're going to start on module 2.3, which is nucleic acids. Thank you all very much for watching, and I'll see you in the next video. Goodbye.